Bom, pessoal, sejam bem-vindos a mais uma palestra do nosso ciclo Ambihidro 2020, do nosso doutorado profissional em modelagem e tecnologia para meio ambiente aplicadas em recursos hídricos. O nosso palestrante de hoje é o professor Ricardo Bello-Mendoza, que é engenheiro químico formado pelo Instituto Tecnológico de Tapachula, no México, com mestrado e doutorado em engenharia química na área de tecnologia ambiental pela Universidade de Manchester, no Reino Unido. Foi pesquisador sênior no Departamento de Biotecnologia Ambiental do Colégio de la Frontera Sur, em Tapachula, no México, de 2000 até 2013, e desde 2013 está na Universidade de Canterbury, na Nova Zelândia, onde atualmente é professor sênior. A palestra que o professor Mendoza irá nos apresentar hoje é intitulada Bioelectrochemical System for Enhanced Wastewater Treatment and Energy Recovery. E essa palestra está relacionada com a linha de pesquisa Tecnologia para o Meio Ambiente aplicada a recursos hídricos do nosso doutorado. Vamos à palestra do professor Bello-Mendoza.
Hello. In this short presentation, I will describe the working principles of bioelectrochemical systems and how can they be used in wastewater treatment with concomitant energy recovery. Wastewater treatment is one of the most important services that local and regional authorities provide to their communities. Wastewater treatment protects public health and the natural ecosystems. In most conventional wastewater treatment plants, the main goal is to remove organic matter, often also remove nutrients, nitrogen and phosphorus, as well as inactivate pathogens in the final effluent. Most conventional municipal wastewater treatment plants are based on the activated sludge process. This is a biological aerobic process. And as a result of the needs of sustaining the aerobic microbial metabolism, there is a need for supplying oxygen, aerating the vessels where the microorganisms are growing. This is a high energy consuming process and is estimated that about 0.6 kilowatts per hour are required to treat each cubic meter of sewage. And about half of this goes to the electrical energy to supply air for the aeration of basins. So there is a need for optional processes that are equally effective in removing organic matter from the wastewater, but systems that are more efficient in terms of their energy consumption. One of these alternatives are the use of high-rate anaerobic reactors. These are also biological processes, and the main difference is that in this case microorganisms don't need oxygen to sustain their metabolism. So these are anaerobic biological processes. And actually in this process for archaea, strict anaerobes, oxygen is actually toxic. So we don't need to invest energy for constantly supplying oxygen for these processes. And actually, as a result of the anaerobic metabolism, the initial organic matter contained in the wastewater, sugars, proteins, etc., cellulose, they can be degraded down to a mix of gases that are called collectively biogas. And biogas is made up of methane, 50 to 60% is methane, 30% is carbon dioxide, and the rest is small amounts of other gases like hydrogen, hydrogen sulfide, methane, etc. Now, due to the high content of methane in biogas, this is a flammable gas, so it is a natural energy carrier. So the anaerobic processes can have a positive energy balance. They do not require oxygen to remove organic matter from the wastewater, but at the same time they can produce energy in the form of methane. One of the most conventional and most common anaerobic processes used, especially at high scale, industrial scale, is what is called the upflow anaerobic sludge blanket reactor, or UASB reactor. And the basic principles of the UASB reactor is that we can grow the microorganisms in a self-attached biofilm. This is a granule, and granules have a density that is slightly higher than that of water, and because of that they can settle at the bottom of the reactor. The picture here at the top is the a photograph of the granules that we cultivated in the laboratory. The picture at the bottom shows electron scanning a micrograph of the same granules. So we can see this is a dense community of microorganisms where the initial organic matter, the substrate in the wastewater, will be eventually converted into carbon dioxide and methane. Now, the UASB reactors work by promoting the growth of these granules within the reactor. The raw wastewater is fed from the bottom of the reactor, and it goes up in an upward fashion. In this way, the water will be passing through a bed of sludge, which is represented here by these orange circles, and the microorganisms then will degrade the organic matter in the wastewater. As a result of this, biogas will be produced, and at the top we'll be collecting this biogas, and the biogas can be sent to and be converted into electricity using a generator. In the very top we can then separate also solids. The solids will eventually settle, and the effluent, the supernatant, will be collected in weirs, and this will be the treated effluent coming out of the anaerobic reactor. So again, this is a process that is quite effective in removing organic matter. We can achieve, well, in the laboratory we can achieve 90-95% removal of BOD or COD. This is biochemical oxygen demand, or chemical oxygen demand. In real scale plants, usually the efficiency is lower. In the case of municipal wastewater, we quite often see efficiencies of 60-70% COD removal. Now, something important to notice is that UASB reactors are being used for the treatment of municipal wastewater, but this is usually happening in developing countries. In China, in Brazil, there are a large number of UASB reactors operating, in Colombia, in Mexico, in India, etc. In industrialized countries, the UASB reactors are mostly used for the treatment of industrial wastewater, mostly from the food industry and the chemical industry. Now, there are different chemical and biological processes that can be used to harvest energy from the organic matter contained in wastewater. In this case, we identify organic matter as biomass. And as mentioned before, anaerobic digestion is a way to collect or recover energy from the biomass. We can then convert this energy, or this carbon, into methane, which is an energy carrier. This can be converted into electricity using a generator. That's one way. But the other possibility is that we can also use the microbial fuel cells. Microbial fuel cells are also biological processes in which the organic matter will be directly converted into an electric current. There is also the possibility of, when we are actually applying an external potential, we can produce hydrogen from the biomass, and this is what we call the microbial electrolysis cell. Now, to understand the potential for energy production from these biological processes, we can look at the flow of electrons during the respiration process. This is the electrochemistry of the process. Now, we have to keep in mind that all these reactions happened because we are coupling a reaction that will produce electrons with a reaction that will take up these electrons. When we put these two reactions together, we form what is called a redox reaction, an oxidation reduction reaction. So the organic matter contained in the wastewater will potentially produce is a source of electrons during the oxidation processes, and this is energy. At some point, well, and this substrate is a mix of different molecules, proteins, cellulose, sugars, carbohydrates, etc. But at some point, these electrons will be carried by a cofactor, and this is the NAD cofactor, the nicotinamide adenine dinucleotide molecule. So this is the source of electrons, and for this process to happen, we need to have an electron acceptor. So during respiration, the electron acceptor is the oxygen. The molecule of oxygen will be then reduced to water. Now, each of these half reactions have different potentials. The potential in the substrate, the electron donor, is usually negative, so there is potential for releasing energy, whereas in the case of the potential of the electron acceptor, in this case the oxygen, the potential is positive. Now, if we couple these two reactions, then there will be a flow of electrons from the donor to the acceptor. And the further away these two reactions are in this tower, so this is like a tower, the more separate these two reactions are, the more energy will be produced. So if we couple these two reactions, say glucose, for example, which is a molecule with a high content of energy, the combination of glucose with oxygen, which is a very strong oxidant, will produce an electron potential of 1.14 volts. There is a lot of energy that can be harvested from these two reactions, and microorganisms that use oxygen in their metabolism and are growing on glucose, for example, they will usually grow at a very fast rate because they are harvesting a lot of energy from the substrate. Now there is the possibility of not using an external electron acceptor, this is not using oxygen, and instead capturing the electrons in an anode. So that means that there will be a potential of the electrons that can be transferred from the substrate to the anode, and this is the potential for the bacteria that are mediating these reactions. Now, also these electrons will eventually be transferred to a cathode, and altogether this will be the potential of the microbial fuel cell. Also, because this process is not perfect, is not 100% efficient, we can also expect some loss of potential in this process, and this is usually related to the internal resistance of the system. So one way to reduce this loss can be maybe using, optimizing the design of the microbial fuel cell, maybe using different metals to transfer the electrons, also using shuttles, buffers in the cathode to improve the transfer of electrons, etc. So microbial fuel cells work by coupling, or we observe the electron donor and the electron acceptor reactions happening in two different separate chambers. In one chamber, which is the anode, we provide the food for the microorganisms, and the fuel for this process are the waste that we want to treat, to stabilize. So the waste will then release electrons. The electrons in this case will be captured in the anode, and the electrons then will be transferred to the cathode via an external circuit. So we use a resistant here, and the change in potential, the drop in potential, will be then the current, the electric current that we can potentially harvest from this microbial fuel cell. In the anode, the electrons will be accepted by oxygen, so we provide oxygen, and the oxygen then will be reduced to water. On the other hand, we have these two cells separated by a membrane. This is a proton permeable membrane where protons can be transferred from the anode to the cathode, and they will be then participating in the reduction of oxygen. As a result of this, there is a current that can be harvested from the microbial fuel cell. Now, a way to actually achieve this in the lab, and some of the earlier applications that we found, is to connect two different chambers, two different bottles, via a proton exchange membrane, which is happening here. Now, in the cells, we can use iron cathodes. We can also use carbon cathodes and anodes, and we usually provide oxygen in the cathode in order for this reaction to happen. Now, how do electrons actually reach the electrode? There are at least 3 known mechanisms for this to happen. One possibility is that the electrons are actually transferred from this microbial cell to the surface of the electron via a mediator, such as the biocyanin produced by Pseudomonas. So that means this molecule acts as a shuttle to transfer electrons from the surface of the cells to the surface of the anode. One possibility is that the microorganisms, the cells, actually get together close enough to the surface of the anode so that the electrons can directly be transferred from the outer membrane onto the electron anode cell. And the other potential mechanisms that has been observed in some cases is the use of conductive villi. This is some bacteria can actually produce some protuberances, some wires that will connect the cell to the surface of the electrode. And this has been observed. Well, this is an scanning electron micrograph showing some cells with the villi that they are producing. So these are potentially the conductive wires for the electrons to transfer from the cell to the surface of the electrode. Now, some genuses of bacteria that has been observed to produce this villi are Geobacter, Alteromonas, and Chigunella. Now, there might be other mechanisms maybe still unknown, still to be discovered for this process to happen. Now, we conducted a set of experiments in the laboratory in order to try to exploit the potential of the two systems, the UASB and the microbial fuel systems. The idea here is that the UASB systems are good, they are efficient in removing organic matter, but they have their limitations. So they usually achieve maybe 70-80% COD or BOD removal, which means the effluent still contains some residuals of organics that needs to be removed in a maybe polishing post-treatment. On the other hand, the microbial fuel systems are more efficient in harvesting energy even at lower concentrations of substrates. So we were wondering whether it might be possible to connect a USB reactor with an MFC so that we can actually produce an effluent of better quality and also improve the overall energetics of the system. This is improve the recovery of energy. So the questions for this particular experiment were, does the MFC actually improve the effluent quality of a USB reactor, and does it improve the energy recovery? So these are the questions that we had. So we conducted this set of experiments in the laboratory using a small 1 liter volume USB system. We used real domestic wastewater to feed this USB. Over time, we achieved the growth of anaerobic granules within the USB reactor, And the effluent of this USB reactor was then fed in batches to the microbial fuel system. So we were using two separate chambers connected by a proton permeable membrane. The effluent of the USB reactor was fed to the anode. We connected the anode with the cathode via an external circuit with resistance here. We were measuring the voltage produced from this current, and in the cathode we aerated, so we provided the oxygen required for the reduction of oxygen to happen, so to capture these final electrons. So very in brief, the main results that we found is that, well, we conducted this experiment with different retention times, hydraulic retention times. This is how long the water remains in the system. So we started with short retention time of 3 hours, which in the case of a USB reactor is actually short. And still we found that the removal of total organic carbon was reasonably good, so something around 50%. We then increased the retention time from 3 to 6 hours, and from 6 hours to 12 hours. And we found that a longer retention time produced a better quality effluent. This is the removal of carbon in the effluent was higher, and it was getting close to 70% for an hydraulic retention time of 12 hours. We did the same with the microbial fuel cell. In this case, again using the raw wastewater with a short retention time at the beginning, 6 hours, and then we move to 12 hours. And similarly to the USB reactor, when we increased the retention time, we improved, or the quality of the effluent improved, so there was a higher efficiency in the removal of organic carbon. Now the interesting part was to see what will happen when we connect the USB reactor with the microbial fuel cell. So what we found is that by coupling the USB and the MFC systems, each of them with a 3 hours retention time, we got a very reasonable removal of total organic carbon, something close to 70%, which is good for a 6 hour retention time overall. When we increase further the retention time using a combination of 3 hours HRT in the USB reactor and 9 hours retention time in the MFC, the efficiency of removal increased slightly. And when we change the combination, it's still 12 hours total retention time, but now with 6 hours in the USB and 6 hours in the MFC, the removal of total organic carbon was even better. And actually this is the highest removal efficiency that we observe. So one question that we were answering here is that actually the combination of USB and MFC system allowed the achievement of a higher removal efficiency than the use of the USB reactor alone. So this is the system that will be comparable to this system. So 12 hours in the USB, 12 hours in the total system, in the combined USB MFC system. The other interesting observation that we found is that when we used a combination of USB and MFC, the system was more stable, and we can see this in terms of the standard deviation. So in the case of the USB, the mean removal efficiency was around 70%, but this was highly variable. Sometimes we achieve very, very good efficiencies, close to 90%, but sometimes the efficiency also dropped to about 40%. The combination of the USB and the MFC system allowed for a system that was more stable. So this is a narrower standard deviation, which is also interesting in terms of how consistent the quality of the effluent can be. Similarly to the removal of carbon, we observed similar trends in the case of the removal of total suspended solids. These are the solids that we can observe in the wastewater and the solids that produce turbidity in the water. So when we started using a retention time of 3 hours in the USB reactor, the removal of TSS was not very high, which is reasonable. 3 hours is very short, so the water passes through very quickly, and there is not enough time for the microorganisms to degrade, to hydrolyze the solids in the water. When we increased the retention time from 3 hours to 6 hours, there was an important improvement in the removal of TSS, and this improved slightly more when the retention time was increased to 12 hours. Still, 12 hours retention time for a USB reactor is relatively short. Similarly, in the case of the MFC, this is a batch reactor. The USB was a continuous process. The MFC is a batch reactor. We observed that by increasing the retention time, the removal of total suspended solids also increased. But then when we combined the USB and the MFC, the removal of TSS was even better. And similarly to what we observed with the TOC, the total organic carbon, the system was more stable. The standard deviation was narrower. So far, we saw that there is potential for improving the quality of a USB reactor if we use a microbial fuel cell. The next part of the study was focused on actually understanding the efficiency of the microbial fuel cell under these conditions. So one way to look at this is to see how much electricity, what is the voltage that we can produce from the microbial fuel cell. This graph is showing the voltage that happens at different cycles. This is because this is a batch process. We cannot expect that the voltage will go up initially and then it will be dropping as the substrates are being used in the anode. So there is a decline in the concentration of food for the microorganisms. When we inject a new batch, then again the voltage goes up and then goes down and so on and so forth. Now, just for comparison, we can see that the voltage that we found in this microbial fuel cell was small compared to other systems. We got a voltage of 0.12 volts, whereas in the case of glucose, other authors have reported a voltage that is more than twice that value, 0.5 volts. That is in the case of a system where there is no proton exchange membrane. But even when there is a proton exchange membrane and an increased internal resistance in the system, the power density in this case has been higher than the one that we achieve. And this is understandable. Glucose, as mentioned before, is a substrate that contains a high level of energy, so the microorganisms get a lot of energy from degrading glucose, especially when the initial concentration is high. In the case of the domestic wastewater, it is usually a very diluted waste, so the content of organics is much lower. And it's also quite often there are solids that need to be hydrolyzed before the molecules are actually available for the microorganisms to produce the energy. Now, looking at the performance of the microbial fuel cell, we saw that the highest power density, this is the highest energy level that we could achieve, was obtained when we were using the raw wastewater. And again, this is understandable because the concentration of substrates was higher. In the case of the effluent from the USB, the concentrations of substrates is lower, usually is some residuals of volatile fatty acids. And then as a result of this, there is less energy being produced. Also, the retention time can play a role in the power density. The Coulombic efficiency is a measure of how much energy is actually collected from the substrate compared to the potential energy, the theoretical energy that can be obtained if all the substrate is converted to electricity. Next, just to characterize the MFC, we conducted a set of experiments to assess how the voltage in the cell changed with the resistance, the external resistance, and the load that we were applying. Because we didn't have potentiostat in the lab, what we did was to to change the resistance manually, increase the resistance, and we ran a set of experiments to assess how the voltage changed with resistance. And this is the kind of curve that we got. Now from this curve, from the voltage, we can calculate the current using Ohm's law. And once we have the current, we can then calculate what is called the polarization curve. The polarization curve is shown here with the red dots. And this is basically showing how the cell voltage changes with the current density. Often in microbial fuel cells, we obtain linear zero-order kinetics for the polarization curve. In our case, the kinetics of the electrochemical system was more close to a first-order reaction, which means we couldn't calculate the internal resistance of the system. And what this curve is telling us is that initially the activation losses seems to be the main factor in the drop in voltage, and from one point, a given point, is the ohmic resistance that is playing the most important role. Now, from this polarization curve, then we calculated the power density curve, which is shown here in the blue dots. So this power density curve is telling us that when the system is open, here at 0, there is no current, and from this point, as we increase the density, the current density, there is an increase in the power density until a point is achieved where the maximum power density is observed, and the modeled maximum point that we obtained was something around 25 milliwatts per unit area in the anode. This is per square centimeter. So from this point, usually the power density starts to decline due to ohmic losses, which is also related to this part of the polarization curve, and also to overpotential in the electrodes. And if we continue this, there will be a point in which there will be no power density observed once the system is in short circuit. So these are curves just that can help us to characterize the efficiency of the microbial fuel cell. And what we did here is to see how the COD removal efficiency, the removal of organic carbon in the water, changes as we change the external resistance in the system, and how also the power density changed. And we found that using an external resistance of 1000 ohms, we could obtain the COD removal efficiency of 80%, which is reasonable for this particular type of wastewater, real domestic wastewater, with a power density of 21 milliwatts per square meter of electrode. So the conclusions of this short project experiment is that the coupling of a USB reactor with a microbial fuel cell can produce a system that produces an effluent of a better quality than the two reactors operated in separate. Okay. The other observation here is that we could produce up to 173 milliwatts per square meter in the microbial fuel cell. There is still a need for making a complete energy balance in the system. We were able to keep track of the current, the power being generated in the microbial fuel cell, but we were not able to capture, especially not in an accurate way, the production of methane in the USB reactor. And this, I suspect, is due to the fact that the domestic wastewater has a low concentration of substrate, so there is a low production of methane, and also there is a loss of methane because it gets dissolved in the water and it goes out of the system with the effluent, so something that is difficult to measure. And maybe also we had a system that was not completely tight, so maybe there were some losses. So there is still a need to actually see how much energy can be recovered from the system, especially because there is a need for aeration in the cathode. So we need to see whether the energy that is being produced in the system is actually higher than the energy required for the aeration. And also, the optimization of the operational parameters in this system could improve the performance of the system. So there is still room for improving the performance of the UASB MFC system by changing some of the and optimizing the design of the system. Now, what we want to do now with the help of a recently recruited PhD student is to explore the possibility of combining anaerobic digestion with the concept of microbial electrolysis cell to produce a biogas that is with higher content of methane. So microbial electrolysis cells work similarly to what we observed before in the case of the MFC. So we have two chambers, one anode and one cathode. The anode is where we feed the waste to the system. So this is an anaerobic system. There is no oxygen in the anode. The bacteria will degrade the substrate. Electrons will be produced. These electrons will be captured in the anode, and the electrons will be flowing through the cathode via an external circuit. Protons will be transferred from the anode to the cathode via a permeable membrane. And in the cathode, the oxygen will be then reduced to water. Now, if we actually apply an external power, so we have a power supply to the system, then we can achieve the electrolysis of the water in the cathode, and as a result of this, there will be formation of hydrogen. Now, the advantage of coupling this in a microbial electrolysis cell is that there is a reduction in the power that is needed to achieve the production of hydrogen. So we might be needing 0.25 volts to produce the hydrogen, whereas in a conventional water electrolysis cell, the need is of 1.8 volts. So there is a reduction because some of the energy is actually coming from the anode in this system. Not all the energy is coming from externally from the power supply. Now, if we actually have a system that doesn't require oxygen, which is an advantage, so there is not a need for aeration, we have an anaerobic cell, and we then remove the membrane, there is potential for the hydrogen that is being produced in the cathode to be used by hydrogenotrophic methanogens. This is methanogens or bacteria that will produce methane out of hydrogen and carbon dioxide. And as a result of this, we might have biogas that has a lower content of carbon dioxide and a higher content of methane. So biogas with a higher content of energy overall. So this is what we want to explore now in the lab and see whether this can actually improve the recovery of energy from wastewater. Okay, so I think that's all for my part, and thank you for your attention.